welcome to Medical Dialogues, your daily dose of health and medical news. I'm Mr. Zaman and here is what I bring to you all from the world of medicine. Possible dispersal of covert aerosol droplets through wind instruments. In Physics of Fluids by AIP Publishing, researchers from the University of Pennsylvania worked with musicians from the Philadelphia Orchestra to deepen our understanding of how much aerosol is produced and dispersed by wind instruments. The researchers used visualization to characterize the flow and then tracked fog particles in the air with a laser. They also measured aerosol concentration from wind instruments with the particle counter. Then they combined these two measurements to develop a simple equation to describe aerosol dispersion. Aerosol speed decays with distance from the instrument. The idea is to help other researchers determine how far aerosols will travel by measuring the exit flow speed. This informs how fast the flow will decay. Aerosols emitted by wind instruments shared a similar concentration and size distribution compared to normal speech and respiration events. We were surprised that the amount of aerosol produced is of the same range as normal speech, said author Paulo Aratia. I was expecting much higher flow speeds and aerosol concentrations. Flow measurements show that exit jet speeds are much lower than coughing and sneezing events. For most instruments, the maximum decay length is less than 2 meters from the instrument's opening. Consequently, wind musicians should stay 6 feet apart, similar to the recommendation for individuals. How the brain gathers threat cues and turns them into fear. Sark scientists have uncovered a molecular pathway that distills threatening sights, sounds, and smells into a single message: be afraid. A molecule called CGRP enables neurons in two separate areas of the brain to bundle threatening sensory cues into a unified signal, tag it as negative, and convey it to the amygdala, which translates the signal into fear. The research published recently in Cell Reports may lead to new therapies for fear-related disorders such as post-traumatic stress disorder or hypersensitivity disorders such as autism, migraines, and fibromyalgia. The team conducted several experiments to test their hypothesis. They recorded CGRP neuron activity using single-cell calcium imaging while presenting mice with multi-sensory threat cues, enabling the researchers to pinpoint which sensory modality involved which sets of neurons. They determined the path the signals took after leaving the thalamus and brainstem using different colored fluorescent proteins, and they conducted behavioral tests to gauge memory and fear. Taken together, their findings show that two distinct populations of CGRP neurons, one in the thalamus, one in the brainstem, project to non-overlapping areas of the amygdala, forming two distinct circuits. Both populations encode threatening sights, sounds, smells, tastes, and touches by communicating with local brain networks. Finally, they discovered that both circuits are necessary for forming aversive memories, the kind that tells you, stay away. Combining antiviral drugs and antibody therapy to treat seasonal flu. Researchers at McMaster University have found a class of well-known antiviral drugs could be part of a 1-2 punch to treat seasonal influenza and prevent a flu pandemic when used in combination with antibody therapies. Antiviral drugs such as Tamiflu have been prescribed for decades to treat flu symptoms in people at risk for serious complications. Researchers found when these medications were used with antibody therapy, the combination was more effective than either approach alone. The antibodies were significantly more efficient at killing infected cells, and the drugs were more potent. The findings published in the journal Cell Reports Medicine could inform new approaches to protecting high-risk groups, including the elderly and children, during an emerging influenza pandemic, the researchers say. The team has studied broadly neutralizing antibodies which fend off a wide range of respiratory viruses for over 10 years. They are examining how these antibodies could be trapped to protect against all strains of flu in their urgent pursuit of a universal flu vaccine. Antibody therapies were used to treat COVID-19, and in theory they could be used to treat flu as a new therapeutic approach, says Matthew Miller, a lead author of the study. For the study, which was conducted on mice, researchers combined antibodies with antiviral drugs. They found the drugs improved the virus-fighting properties of the antibodies, which work by binding to the surface of an infected cell, then triggering our immune system to kill the cell before the virus can spread. Researchers also report using a combination therapy may extend the life of current antiviral drugs because viruses are less likely to become resistant to such drugs when delivered in conjunction with an antibody therapy. pre-fertilization DNA transfer to avoid mitochondrial disease inheritance. Transferring the nuclear genome from one egg into the cytoplasm of a donor egg is a strategy to enable women carrying mutations in their mitochondrial DNA to have healthy babies. A new study published in the open access journal PLOS Biology uses multiple omics technique to show that this strategy called spindle transfer is likely to be safe with little evidence of genetic or functional difference between the resulting embryos and healthy controlled in vitro fertilization or IVF embryos. The results are likely to spur further adoption of spindle transfer for IVF when there is a risk of mitochondrial disease. The spindle refers to the division apparatus that holds the nuclear chromosomes in suspension until fertilization. During spindle transfer, the maternal spindle is removed from an unfertilized egg and placed into a donor egg that had its own spindle removed. Spindle transfer has been used clinically, but there remains question about its safety. To shed light on this question, the authors performed 3 different types of analysis on single cells from 23 blastocysts following spindle transfer and compared them to 23 control IVF blastocysts. They found no difference in DNA copy number, a measure of genomic integrity, between spindle transfer and control blastocysts. RNA expression profiles were also similar between the 2 blastocyst types, regardless of which layer of the blastocyst the cells were taken from. The authors did find a small but significant reduction in the level of DNA demethylation in spindle transfer blastocysts in one layer, trophectoderm, though not in the 2 other layers. DNA demethylation is one of the processes used to increase gene expression during development, and their analysis suggested that the reduction was evidence of a slight delay in the process rather than a permanent inability to upregulate the affected genes. That's all for today. Stay tuned to Medical Dialogues for latest updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.